Skin color is one of the first things we see. But what lies beneath that variety? In this film, I'll show how genes, sunlight, environment, and migration shaped skin tones among human populations. We'll explore how pigmentation works, melanocytes, melanosomes, types of melanin, and how they act. We'll examine how pigment is made, how it moves, and what factors regulate it. We'll address adaptation to UV radiation and how the environment sculpted pigmentation. We'll show human migrations and how skin colors changed depending on where people lived. This is only the beginning. Later, we'll dive into genes, adaptations, and social impact. Let's begin. To understand skin color, we must first look under the surface, the structure of skin, and how pigment cells work. Melanocytes are specialized cells in the lower layer of the epidermis. They synthesize pigment and transfer it to other skin cells. Melanosomes carry melanin pigment into nearby keratinocytes. Skin color depends more on melanosome number, size, and distribution than just cell count. There are two main types of melanin: eumelanin, brown to black, and pheomelanin, yellow to red. The balance between them influences skin tone. Melanin synthesis, melanogenesis, is a multi-step biochemical process. Enzymes such as tyrosinase and regulators like AMSH guide which pigment is produced. Hormones, signaling molecules, and environmental cues regulate pigment production. They can upregulate or downregulate melanogenesis. So skin pigmentation arises from a complex network: cell structure, pigment synthesis, molecular regulation, all working together. Ultraviolet. UV radiation from the sun plays a central role in shaping skin color across evolution. While UV triggers production of vitamin D, it also degrades folate, an essential nutrient for DNA synthesis and fetal development. Excessive UV exposure can damage DNA, increase risk of skin cancers, and cause mutational stress. UV intensity varies greatly by latitude. In high UV regions near the equator, darker skin offers strong protection. Skin must balance photoprotection with vitamin D synthesis. Too much pigment blocks UV needed for vitamin D. Too little increases UV damage. In the skin, UVB converts 7-dehydrocholesterol into provitamin D3, then into active vitamin D3, critical for bone health, immunity, and more. These evolutionary pressures, folate protection and vitamin D synthesis, drove selection of skin pigmentation across environments. Modern humans originated in Africa, then migrated into new lands with varying UV exposure. As humans traveled, they moved into latitudes with lower or more seasonal UV, shifting selective pressures on skin color. Populations in equatorial zones retained darker skin. Those in higher latitudes often evolved lighter skin to allow UV penetration. Multiple waves of dispersal, coastal migration, inland routes, each carried genetic variation and adaptation potentials. Over tens of thousands of years, natural selection, genetic drift, and gene flow shaped skin color variation globally. Where populations met and interbred, skin color traits mixed, creating gradients rather than sharp boundaries. Migration, environmental variation, and genetic exchange all acted in concert to produce the wide palette of human skin tones we observe today. Skin pigmentation is a polygenic trait. Many genes contribute to your skin tone. Genes such as MC1, R, TYR, OCA2, and SLC24A5 are among the most studied. Different alleles influence melanin levels and type. For example, the SLC24A5 gene variant, ALA111THR, accounts for a large proportion, 25 to 40% of the difference in melanin index between Europeans and West Africans. Many variation differences are regulatory rather than coding. Enhancers, promoters, SNPs, outside gene bodies influence expression. Different populations independently evolved lighter skin via different genetic paths. Convergent evolution in adaptation to low UV. Recent studies identified up to 135 genes influencing pigmentation, many previously unknown regulators and modulators. This complex network, combining structural genes and regulatory elements, shapes the skin color spectrum we see today. Some populations challenge simple rules. Inuit, living in low UV zones, present interesting pigmentation dynamics. Their diet rich in vitamin D, fish, marine mammals, may buffer selection for lighter skin. Near the equator, intense UV meant selection for darker skin persisted, balancing protection and function. High-altitude populations also adapt pigment and physiological responses to UV intensity variations at elevation. Cultural, behavioral factors, clothing, shelter, outdoor habits, moderate UV exposure, and influence selection. In admixed populations, combinations of pigmentation alleles from distinct ancestries create unique skin tone distributions. By studying exceptions and mixtures, we better understand how flexible and context-dependent pigmentation evolution is. Adapting skin pigmentation involves trade-offs. Protection versus UV absorption for vitamin D. Dark skin reduces UV damage and mutation risk. Light skin allows more UV penetration, but carries higher skin cancer susceptibility. Too little pigment can lead to vitamin D insufficiency in low UV areas, affecting bone health, immunity, and more. In regions with strong UV, darker pigmentation protects. In weak UV, lighter skin helps absorb necessary rays. Modern health outcomes often reflect these evolutionary legacies. Skin cancer rates, vitamin D deficiencies vary by pigmentation. Medical technology sometimes fails darker skin tones. AI diagnostic tools have worse performance for dark-skinned patients. Understanding these trade-offs helps us appreciate both evolutionary history and current health disparities. Modern genomics is revealing new pigment-related genes and regulatory mechanisms at unprecedented detail. A recent genome-wide screen tied melanin concentration to light-scattering properties of pigment cells, confirming functional links. Scientists have identified new regulators like KLF6 and COMMD3 that adjust pigment synthesis via melanosome acidity and control. Many pigmentation variants act through regulatory DNA rather than altering protein sequence, changing when and where genes are active. Tracking allele frequency changes over time helps map historical pigment adaptation in various regions. Functional experiments in cells and animal models test how variants affect melanin production and response to UV. These new genetic insights refine our understanding of skin color as a dynamic, multi-layered trait. Color of skin has long been misused as a basis for hierarchy, discrimination, and pseudoscience. But biologically, pigmentation is an adaptation, not a moral or intellectual metric. Stereotypes, bias, and unequal treatment still persist based on skin tone. Education and science help correct these myths. As mentioned earlier, AI models often underperform on darker skin images, a modern manifestation of bias. Skin color varies continuously. Human diversity is a gradient, not discrete boxes labeled race. Understanding the science helps us see unity across diversity. We share far more than we differ. Let's move forward grounded in science, respect, and curiosity, rather than division. Skin color is a living record of human evolution, built by genes, sunlight, migration, and adaptation. Each factor, pigments, environment, movement, combine to create the vast spectrum of human skin tones. Modern genetic research continues to reveal hidden complexity in what seemed a simple trait. These evolutionary legacies carry into present-day health. Pigment variation impacts UV risk, vitamin D metabolism, and medical equity. Let's rethink how skin color is framed, not as division, but adaptation, variation, and shared humanity. If you found this enlightening, share it, ask questions, dive deeper. Science is our tool to understand, not separate. Thank you for watching. The story of our skin is the story of us all. Share your comments below